actually happens at the boundary between two neurons, between the axon of one neuron and the dendrite of another. This interface is called a synapse. Most neurons are connected together by a much more complicated structure called a chemical synapse. In a chemical synapse, rather than simply passing along an electrical signal from one cell to another, the action potential travels to the end of the axon and causes a chemical to be released into a very small space between the two neurons called the synaptic cleft. This chemical is taken up by the downstream neuron on the other side of the cleft. This chemical signal can cause the downstream neuron to depolarize its membrane, converting the chemical signal back into an electrical one. Or it can have other effects on the cell. This chemical step is slower than transmission across an electrical synapse, but it opens up an enormously diverse repertoire of different and more complex kinds of signaling. And synaptic function plays a critical role in computations performed by neurons. We'll spend the rest of this unit exploring the inner workings of chemical synapses, on our way to beginning to look at how networks of interconnected neurons give rise to behavior.
Everything you see here is real data. We're looking through a microscope at neural tissue, going down through an area of the brain called the medial prefrontal cortex. We can use these microscopic images to reconstruct the three-dimensional layout of the neurons and really see the intricate and intertwining patterns that they make. These connectivity patterns throughout the brain underlie all neural function and are responsible for everything from controlling your muscles when you tie your shoes to how you feel about your family. Understanding these patterns is a massive ongoing project for neuroscientists everywhere, and could lead to breakthroughs for everything from consciousness to depressive disorders and schizophrenia. In this example, we injected purple dye into a far distant brain region called the amygdala, which is largely where our emotions and feelings of fear are generated. The purple wires are axons, or connections, coming from the amygdala into this area. We've also used some recent advancements in genetic engineering to select only the neurons inside this region that connect to the amygdala and dye them green. By combining both the green and purple dyes, we're able to see the full circuit of connections, viewing both the neurons that connect to the amygdala and connections coming back in from the amygdala. The locations and patterns of the neural connections, called synapses, show us how heavily these two groups of neurons are connected. To help us start to understand the incredible complexity of these neural networks, we develop computational techniques that segment or isolate individual neurons. By isolating a neuron, we can measure its characteristics, like size, shape, how many branches it has, and what general type of neuron it is. This may not seem like much, but it's really important for understanding so much about the brain.